Have you ever really thought about what the instruction manual for every single living thing on Earth looks like? Well, it's called DNA, and it's packed into pretty much every cell in your body. Today, we're going to deconstruct this incredible molecular blueprint and see exactly how it builds and, crucially, copies itself. Okay, let's dive right in.
So, here's our game plan. First, we'll set the stage with the science behind all this. Then, we're going to zoom way in on that famous double helix structure. After that, you'll meet the amazing enzyme crew that does all the heavy lifting. We'll then walk through the step-by-step process of making a perfect copy, and finally, we'll zoom back out to see why this tiny molecule has such a massive impact on, well, everything.
All right, first up, the blueprint of life. We're starting big picture here, looking at the whole field that studies these fundamental codes of life.
The field we're exploring is called molecular biology, and really, it's exactly what it sounds like. It's all about how life works at the molecular level. We're talking DNA, RNA, proteins. You know, the nitty-gritty stuff. It's all about figuring out how the instructions for life are written, how they're read, and how they get passed down from one generation to the next.
Now that we've got the big picture, let's zoom in. Way in. In this next part, we're going right inside the double helix to learn how that genetic code is actually written.
And there it is, the icon of biology, right? This twisting ladder, the double helix. It was famously figured out by Watson and Crick back in 1953, and I'm not exaggerating when I say this discovery changed everything. It just blew the doors wide open for understanding genetics on a whole new level.
So what is this famous ladder even made of? Well, the basic building blocks are things called nucleotides, and each one has 3 parts. You've got a phosphate group and a deoxyribose sugar. Together they make up the strong backbone, kind of like the sides of the ladder. But then you've got the really important part, the nitrogenous base. These are the rungs of the ladder, and they come in 4 flavors: adenine, thymine, cytosine, and guanine, or as we'll call them, A, T, C, and G.
Now here's where the magic happens. These bases don't just pair up randomly. Nope, there is a strict and honestly beautiful rule they follow. Adenine, or A, always pairs with thymine, T, and cytosine, C, always pairs with guanine, G. That's it, A with T, C with G. This incredibly simple rule is the foundation for all the breathtaking complexity of life. It's the 4-letter alphabet that writes the story of everything from a tiny bacterium to a giant blue whale.
So, we know the structure, but how does a cell make a copy of billions of these base pairs without messing it up? Well, for that, you need to call in the replication crew, a highly specialized team of enzymes that gets the job done perfectly. Think of this whole process like a molecular construction project. First up, you've got helicase. We'll call it the unzipper, because its job is to unwind and separate the 2 DNA strands. Then comes primase, the primer, which lays down a little landing strip to get things started. The star of the show is DNA polymerase, the master builder. This guy reads the original strand and builds the new one. And finally, cleaning everything up, we have ligase, the gluer, which seals up any gaps and makes sure everything is solid.
All right, we've met the crew. Let's put them to work. In this section, we're going to walk through the incredible step-by-step process of how this blueprint is duplicated with amazing precision. Before we jump in, you have to know this one key idea. The process is semi-conservative. It sounds technical, but it's actually a really elegant concept. It just means that when DNA is copied, you don't make 2 totally new molecules from scratch. Instead, each new double helix is a hybrid. It keeps one of the original strands and uses it as a perfect template to build one new strand. It's a genius way to make sure you get a faithful copy every single time.
The whole show can be broken down into 3 main acts. First, there's initiation, which is all about getting things started. Second is elongation, the main phase where all the building happens. And third, termination, where the job gets finished and polished up. Beginning, middle, and end. Simple as that.
So let's start with Act 1, initiation. This whole thing kicks off when our unzipper enzyme, helicase, gets to work. It latches onto the DNA and starts unwinding that double helix, creating this Y-shaped area that we call the replication fork. This fork is the active construction zone, where all the copying is about to go down.
And this, right here, is where the process gets really clever. See, our master builder, DNA polymerase, has a strict rule. It can only build in one direction. For one of the original strands, we call it the leading strand, this is no problem. The new copy can be built in one smooth, continuous piece. But the other original strand, the lagging strand, runs in the opposite direction. It's like a one-way street going the wrong way. So to copy it, the polymerase has to build it backwards, in little short bursts.
These little pieces it makes are called Okazaki fragments.
Finally, we reach our last step, termination. This is where our gluer enzyme, ligase, comes in for the finishing touches. It moves along that lagging strand and stitches all of those little Okazaki fragments together, making one continuous, complete strand. And what's the end result? Two perfect, identical copies of the original DNA molecule, each one ready to go into a new cell. Job done.
So, we've seen the how, but let's zoom all the way back out for the why. In our final section, we're going to connect this tiny molecular process to its absolutely enormous role as the foundation of, well, all of life and a ton of modern science.
So why does this one process matter so much? Because accurate DNA replication is fundamental to literally everything. It ensures that when your cells divide, your genetic information stays the same. It's what allows you to grow up from a baby and for your body to repair itself, like when you get a paper cut. It's the absolute basis of reproduction. And when tiny mistakes or mutations do happen, it becomes the source of all genetic variation which drives evolution itself. This process is the bedrock of entire fields, from medicine and genetics to forensics.
We've seen just how incredibly precise this molecular machinery is. It's designed to make perfect copies over and over and over again, billions of times. But perfection isn't always possible in biology. So that leaves us with one final and really important question. What happens when this amazing process makes a mistake? Because the answer to that question explains everything from genetic diseases to the incredible diversity of life on Earth.